image a very small handful of cells, whereas on the Mesolens, we can image thousands. Hi, my name is Matthew Keys, and I'm a postdoctoral researcher at the Marine Biological Association. I work in the Wheeler Group, and our focus at the Wheeler Group is the study of the physiology and biology of phytoplankton in the oceans. Imagine a set of binoculars. As you zoom closer and closer in on an object on the horizon gets smaller and smaller, and we refer to that as the field of view. So when we're looking at organisms, microscopic organisms under a microscope, we really have to reduce our field of view the more magnified we get when we want to see a much higher level of detail. The Mesolens microscope works in a very different way. It has a very wide field of view, so when used in combination with a high resolution camera, we can gain very detailed information on multiple organisms across a wide field, and we can zoom into that image once we've captured that image with no loss of quality or resolution of that image. So we're able to get a far more detailed view over a much wider area. So phytoplankton are a very key group of photosynthesizing organisms. They account for somewhere in the region of 50% of global photosynthesis. So we really depend on phytoplankton for removing carbon from our atmosphere and drawing more CO2 into the ocean surface. One group of phytoplankton alone, the diatoms, which are characterized by their silicified cell walls, are responsible for 40% of all photosynthesis in the oceans. In fact, they're responsible for 20% of global photosynthesis. That's as much photosynthesis as conducted by all of the rainforests on Earth put together. So because of this global importance, we're interested in understanding how phytoplankton photosynthesize, their rates of photosynthesis, and some of the physical drivers that can impact photosynthetic performance, all of which lead to differences in the amount of carbon that's being fixed by phytoplankton. We can measure indirectly photosynthesis in phytoplankton simply by imaging and measuring fluorescence produced by chlorophyll. During photosynthesis, phytoplankton are absorbing sunlight, incoming photon energy, that they then convert into chemical energy to drive the whole photosynthetic process. Phytoplankton, like all plants, aren't able to cope with all of the incoming sunlight energy. So some of that energy needs to be dissipated. And in the case of chlorophyll does that by fluorescing or reflecting some of that light back. And we're able to take advantage of that by measuring the intensity of fluorescence that's produced by chlorophyll. So when we're measuring chlorophyll fluorescence, typically we can take algal cells, either from cultures that we grow in the laboratory or from natural seawater samples. And historically, we've measured chlorophyll in closed chamber systems. So we'll have a small detector unit like this that can detect chlorophyll fluorescence. And with a few milliliters of a phytoplankton sample that could contain hundreds to millions of cells, we can detect all of the fluorescence collectively that the cells in that sample are producing. Now that gives us one value of chlorophyll fluorescence, and we use that single value to describe the photosynthetic efficiency of a whole sample. More recently, as chlorophyll fluorometry has progressed, we're able to actually start imaging chlorophyll fluorescence, and that's starting to allow us to delve a lot deeper into some of the chlorophyll fluorescence parameters that enable us to determine and estimate rates of photosynthesis. So when it comes to imaging phytoplankton, because phytoplankton are microscopic organisms, we need to use microscopes in order to conduct imaging, particularly at the single cell level. And over the last 10 years or so, some of these chlorophyll fluorescence measurement techniques have been integrated with microscopes, so we can start to view chlorophyll fluorescence in phytoplankton cells at a single cell level. Because traditional microscopes have a very narrow field of view, That means when we conduct these types of chlorophyll fluorescence analyses, we're only able to image a small handful of cells at any given time. And that makes it very difficult for us to start making inferences about phytoplankton photosynthesis at a population level. The MBA now has a very sophisticated microscope called the Mesolens. There are only a small handful of these currently in existence. The clear difference between traditional benchtop microscopes and the Mesolens microscope is the enormous microscope objective that the Mesolens has. And this enables a very wide field of view. So we're able to view many, many more cells, hundreds to thousands more cells on the Mesolens microscope than we can view on a traditional benchtop microscope. And when used in combination with a high-resolution CMOS camera, we're able to image chlorophyll fluorescence down to a micron scale, where we have somewhere in the region of 1 to 2 microns per pixel. So we're able to capture very detailed images of chlorophyll fluorescence. We apply a protocol of light pulses, very rapid light pulses that excite the chlorophyll. That then makes the chlorophyll fluoresce. That then enables us to say something about the photosynthetic efficiency and the light harvesting efficiency of phytoplankton populations on a per cell basis. So having the Mesolens microscope at the Marine Biological Association enables us to conduct more and more research. We've now spent around about a year taking measurements of seawater samples from the Western English Channel, and that gives us very important information that enables us to say something about which species are contributing more to photosynthesis within a whole phytoplankton population, and how that changes over time when we see these very large shifts in species that tends to occur seasonally. More widely, the Mesolens microscope has been used to examine lots of physiological processes, both in large phytoplankton populations as well as multicellular organisms, anything from calcium signaling amongst phytoplankton populations as well as terrestrial leaves, right down to the opening and closing of stomata in leaves in combination with terrestrial leaf calcium signaling. We're going to be using the chlorophyll fluorescence imaging that we can produce on the Mesolens microscope to conduct a survey of the photosynthetic efficiency of phytoplankton sampled from the Western English Channel right here off the coast of Plymouth. And this is now opening up a lot of new areas in research fields, both internally here at the MBA, as well as our many collaborators who work in marine laboratories around the world who don't have a Mesolens microscope in their laboratory. So it's enabling a lot of future collaborative work in the field of marine research.